Happy Friday, students. Welcome to the second part of heredity, and this should be the last part of our chapter, though I think we'll probably do some kind of activity or lab thing next week, not just jump right into the next chapter, do something that maybe is a little bit more fun for this stuff. So if we're due now here, it says in squirrels, the gene for gray fur, big G, is dominant over the gene for black fur, little g, which means if an animal is going to be black, it has to have two of the little g alleles. If it's going to have gray fur, it could have either one or two of the big G alleles. We don't quite know yet. It says if 50% of a large litter of squirrels are gray, the parental cross that produced this litter was most likely what? So if we want to get 50% gray squirrels, and we know now that gray fur is the big G, we want to see which of these options down here is most likely the cross of the parents. So can we eliminate any choices right off the bat? Anything look weird? I would right away say, what the heck is this uppercase O? I don't know what that's supposed to be. It just says go, go. And I would eliminate that choice right there. Also, if I know that the squirrels I'm looking for are supposed to be gray, and again, we know from right here that gray is the big uppercase dominant allele, you have to have at least one uppercase allele somewhere, right? So I should be able to eliminate this choice right here. So there are no traits, no alleles for gray fur. Not going to be able to do what I wanted to do. So now I'm left with two choices. And look, honestly, I would just go with process of elimination. You know, if I think I'm going to need 50% of the animals getting at least one of the big G's, I'm probably looking for, a combination of alleles where, about half of them are the big G, probably not much more than that, maybe a little bit less than that. So, but I can't be sure. And the only way I'm going to really know for sure is really just to try this. So I'm just going to write these letters out. Let's try the first example. Big G, little g, crossed with little g. Doesn't matter where I write these letters in, so I'm just going to start on the side. I'll do a big G over here with a little g, and a little g up top. Let's see what this ends up looking like and see if this works out. I've got big G, another big G, and then the bottom it would be little g with little g, and a little g with little g. So that sure looks good to me, right? We've got 50% of our animals right here have the big G. That would make them gray. 50% have only little g's, they would end up with black fur. That sure looks like it's going to be our answer. Let's just double check with the other option and make sure we didn't mess up at all. So I'm going to go with choice D, and I'm going to try a big G crossed with a big G, little g. Let's see what this one looks like. I don't think this should work out, but let's just make sure. Top left, we got big G. Top right, it's now big G, little g. Bottom left, big G. Bottom right, big G, little g. So I see three, actually in all four, we're going to get the big G. So yeah, this is not going to work because every one of these options has at least one of the big G's. So every one of these choices will be entirely gray, and that's definitely not the 50% gray we're looking for. So choice B was our answer. Let's see what we're doing today. The other day we looked at the basic ideas of heredity, dominant and recessive genes, and how we can predict traits with Punnett squares. Today we'll see what can be a little off about that, why Mendel was sort of wrong, and how to trace the passing of genes through a family. Calm down, Greg. I mentioned the other day that Mendel didn't know anything about DNA because no one at the time did. Because of that, his ideas are sound but not complete. We now know that there's a lot more often going on than just big letter, little letter models of inheritance. Think of height. Do we have only tall people and short people? If height was controlled by having either a dominant or recessive allele, then everyone in the world would be one of two heights, either tall or short. Maybe you'd get some kind of mixed average in the middle, but typically you'd expect tall or short, dominant or recessive. Does that sound like how it works, though? Of course not. There are people of every height because there's more than just one gene controlling height. Multiple genes produce a wide range of heights in people. So it's not just one thing, it's a couple of different genes working together that give us this complete range anywhere in that bell curve of many, many different heights on people. There are instances of an average trait, though, like I mentioned the possibility of a second ago. So some flowers have alleles that don't show dominant or recessive behavior in the true sense. In this example here, a red flower and a white flower will produce pink flowers. Neither of the alleles is truly dominant. So you've got the red gene, you've got the white gene, but neither one truly wins and overrules the other one. So if the offspring flower gets both a red and a white gene, they will actually show this kind of average or middle ground, and the flower will end up looking pink. And other times we see different coloration within an organism itself, like how a cow has patches of different colored hair. You don't get just a brown cow or a white cow or a black cow. You can get cows that look like this, the traditional thing you probably think of when you think of a cow with those kind of patchy fur colorations. On the same organism within its body, you have different areas, some brown fur over here, some white fur over there. That's all caused by these different genes. You don't have just one thing in one place. You can have different genes kind of isolate on different parts of the organism, and they show up like this. And this is something that's pretty common in nature and even on some people. Sometimes external conditions in the environment can affect how living things' genes are expressed. So the classic example of this is the rabbit. In warmer temperatures, this rabbit's fur grows white, but a similar rabbit raised in colder conditions will have black fur on its feet, tail, ears, and face. The darker fur helps to keep it warm in colder temperatures. That's kind of cool, right? Here's where it gets weird, though. If you were to take one of these rabbits and shave some of the white fur off its back, so you just expose the skin, as you can see there, and then put an ice pack on that exposed spot of skin. And this is not just like for 5 minutes. This is a prolonged thing. You'd keep an ice pack on this poor little rabbit's back on the skin. What do you think would happen? Who guessed black fur will grow where it used to be white? The genes are there for the rabbit to have either white or black fur, but the black fur only actually shows up when it's needed because the outside conditions are cold. So as long as we can get rid of that hair that's there, and now make the body think it needs the black fur to protect it from the cold, this is what will actually happen. That's kind of cool, right? It's sad. Is this the nicest thing to do to some poor rabbit? Obviously not. Don't do this. But it works. And this same picture has been in books and on the region since at least when I was taking the class at Ur. So I always make sure to show this. I think it's cool looking, but obviously, again, don't do this to some poor animal. Love, love, love this cartoon.
Chromia.
A gentleman would at least offer to buy me a drink first.
Norman, pint of bitter for me and a brandy for the lady, please.
How did you know that?
Lucky guess. Name's Xavier. Charles Xavier. How do you do?
Amy.
Heterochromia was in reference to your eyes, which I have to say are stunning. One green, one blue. It's a mutation. It's a very groovy mutation. I've got news for you, Amy. You are a mutant.
First you proposition a girl, and then you call her deformed. How is that seduction technique working for you?
I'll tell you in the morning. No, seriously, though, you mustn't knock it. Mutation took us from single-celled organisms to being the dominant form of reproductive life on this planet. Infinite forms of variation with each generation, all through mutation.
Thanks for that somewhat creepy explanation, Chuck. Mutations happen in cells all the time, but they usually get caught and corrected, so nothing really comes of it. Something to remember is that the only way a mutation can be passed on to the next generation is if the mutation, the change in DNA, is present in an egg or sperm cell, since those are the only cells that contribute to the production of offspring. A mutation in skin cells isn't going to impact someone's kids, but a mutation in an egg or sperm could easily lead to a problem in a baby. When mutations do happen, a lot of times they don't end up causing much of a difference, since many codes give similar results. So for example, in this circle box of the codon chart, all 4 options for the third base in the codon could lead to the same resulting amino acid. So whether it's UCU, UCC, UCA, or UCG, if the change happened to be in that third letter, you're still going to get serine as your amino acid. Some are going to cause much bigger problems much more easily, but plenty of the time, we see also right down here for proline and leucine, same thing. Little changes aren't going to cause any effect. Sometimes mutations do happen. That's a good thing, because it leads to changes that can cause improvements and promote evolution over time. I just said that most mutations don't do anything, and that's mostly because they're often caught and corrected. But when mutations aren't neutral and do have an impact one way or the other, they unfortunately tend to be harmful. This is nature's way of kind of weeding out things that don't work. So this 2-headed turtle, which is almost surely photoshopped, would be a harmful mutation. It's not going to help this thing survive any better, and this trait would not be passed on because the organism would not survive long enough to have offspring. A small percentage of mutations can have a positive effect on the organisms, though, like how these foxes gain different types of coloration to fit into their surroundings better. These don't happen as often, but it's typically important when they do. Mutations can happen in a few ways. So we've seen some examples recently of how letters in DNA change so that the instructions are wrong. That's called a genetic mutation. It's a change in the genes themselves, why it's called genetic, and it's a change in the instructions of DNA. Another kind of change is a chromosomal mutation, which is a mistake in the amount of chromosomes that cells get. If chromosomes are damaged or go to the wrong places during cell division, that would be an example of a chromosomal mutation. In the picture here, the X-shaped chromosome right here doesn't separate properly. It doesn't go where it should during cell division. So right here we had an X that split up, and now one piece goes this way, one piece goes this way. In the example on the left, the X stays together. So that by the time you get now to the next bottom part in the daughter cell on the left, there's an extra chromosome right there, and its sister cell right there, sister daughter cell, has one fewer chromosome than it should have. These two on the right each have 2 chromosomes. This one only has 1, and this one now has 3. So it's causing a chromosome mutation because one cell gets too many of the chromosomes and one cell gets too few of the chromosomes. As for what can cause mutations, sometimes it's simply a typo during the copying of DNA, a proofreading error that doesn't get corrected. Other times, changes are caused by something outside the body. So these influencers are called mutagens. And examples are radiation, including sunlight, any kind of chemical exposure, such as those in cigarette smoke, and radioactive spider bites. We talked about sickle cell anemia much earlier in the year, and this is an example of an autosomal recessive mutation. Autosomal means it's a change on a non-sex chromosome, and recessive because someone has to have 2 copies of the allele to express the trait. So you'd have to get a copy of this gene from both your parents. The mutation causes the crescent moon shape that leads to blood vessels clogging and fewer blood cells. Remember the lab we did, and we talked about this in the blood chapter. There's an extra wrinkle to this story here. Individuals with sickle cell disease actually end up having something of an immunity to malaria because the shape and biochemistry of the sickle cells prevents the malarial parasite from infecting a host's blood cells in the same way that it normally makes people really, really sick. This is a pretty interesting example now of evolution providing a balance between 2 bad alternatives. Someone may have sickle cell disease that's definitely not something you'd want, but at the same time, it prevents a deadly and often just totally disastrous malaria from taking hold. Albinism is another autosomal recessive condition by which 2 copies of a recessive allele lead to a lack of pigment expression in organism. This causes it to look white. So not having color often makes an animal an easy target for predators since it can't hide easily, as well as giving no protection from dangerous solar radiation. As Charlie sort of explained while hitting on some college girl a little bit earlier, heterochromia is a mutation that causes pigment differences. Individuals with albinism, as we just saw, lack pigment, while those with heterochromia will have multiple types of pigment separated on their bodies. This one is a dominant trait, so it's easily passed on to offspring. And usually see it expressed as 2 different color eyes, but sometimes it shows up as a difference in hair or skin coloration too. This is actually not all that uncommon, often as subtle rings or areas of different color in the eyes, so there's a decent chance you know someone who has this and may not even realize it. Some more famous examples include future Hall of Fame pitcher Mad Max Scherzer, actress Kate Bosworth, who you may remember from some such classic films as Win a Date with Tad Hamilton, or this cat who looks like a Batman villain or a two-flavored sandwich cookie. Last mutation I want to show you is called polydactyly. And what do you think this one might be? What's poly mean? But dactyly, that's a much less likely thing that I would expect anyone to know. Do you remember, though, any dinosaurs with dactyl in their names? The pterodactyl, though, one of the flying dinosaurs that you're probably more familiar with, means winged finger. Pterodactyl, winged finger. And poly means many. So polydactyly is when someone looks like this. Polydactyly is a trait that causes there to be more than the typical number of fingers or toes, and it's a dominant trait, so it will be passed on more often than not. This is Antonio Alfonseca, and he was a relief pitcher mostly for the then Florida Marlins, with whom he once led the National League in saves. It's a little tough to see in this picture because of the positioning of his hands, but if we look closely, the coloration on his hands kind of blends together. This picture's not great, but we can see 1 finger, 2 fingers, 3 fingers, 4 fingers, 5 fingers, and right here, the 6th finger is kind of coming out of the picture. It's going towards his camera and kind of blends in, so it's tough to say. And same thing on this side. We've got 1, 2, that was a terrible drawing, 2, 3 is right there, 4 is right here. You can see 5, and finger number 6 kind of blends in. You can see right here like a little kind of coloration. That is the 6th finger coming out of his hand. He's coming towards the camera and it kind of blends in with the rest of his hand, but there are 6 fingers on each hand right there. For the last part of our chapter, we'll take a look at how we can follow the presence of genes and track them through the generations of a family tree. This is a pedigree chart, and it's essentially just a big fancy family tree with information about different traits instead of just showing how people are related. So to read this, circles are females and squares are males. So right here I've labeled with F's and M's, but those are your females if they're circles, and squares are always going to be males. You may notice that some are black and some are white here. The shaded shapes are people who are affected by some gene, so either they have a disease or they show a trait of some kind, and the unshaded ones do not show the trait or disease. Some people who are unshaded, though, may have one copy of the allele but don't have the trait. Like if it's a recessive condition, but the person has one dominant allele and doesn't exhibit it. We call those people carriers since they carry the gene even though it's not able to be expressed. Horizontal lines connecting people show siblings in the same generation or mates. So right here we have a line kind of going between a male and a female. That's showing that these people had kids. The vertical lines go from parents to their kids. So this line now will connect that male and that female to their 3 children. So you see right here, this horizontal line doesn't actually go between the people as it does up here. The horizontal line that just kind of dips down, this is showing that these 3 individuals, 1, 2, and 3 on this level, in this generation, are all siblings of each other. If there was a line between them, like up here, that would, or even right here, that would show that those 2 people had children together. So they're parents of whoever is now extending through the vertical line to the next generation. So just saying to keep in mind as we're seeing this. It's always these kind of rules. If an allele is dominant, we would expect it to show up in every generation at least once. It doesn't need to, but you'd expect it to be pretty common because it is overruling other things. In this example pedigree, we see an affected father who mates with an unaffected woman, producing 4 children, 3 of which have the trait. Now, every kid doesn't have to have the trait because obviously the mother here is recessive for it. So she's only giving a recessive gene. If the kid is recessive for it, that must mean the dad has some recessive gene also, that the kid's then being doubled up for it, and he is not going to have the trait himself. 3 of these kids do have the trait, and the daughter on the right, the last circle right here, then has kids with this man over here who also has the trait like her. So of their 4 children, half are affected with that trait. So again, even though these 2 individuals are both affected, they must have some recessive copy of the trait, because both their, 2 of their 4 children at the bottom then do not have that trait. So they had to have gotten a double amount of recessive traits, double amount of recessive alleles from their parents. On that note, a recessive allele doesn't always show up because it's overruled by a dominant allele and needs 2 copies just to be expressed. That recessive allele is still there though, it's just not visible because it's being suppressed by the stronger dominant allele's influence. An affected parent may not have children with the trait, but it may show up again in the grandchildren's generation. This is where the idea of carriers comes from. A carrier is a person who has a copy of a recessive allele, but it doesn't get expressed because they also have one dominant copy. If 2 of these carriers have a child, a Punnett score will predict a 25% chance that the child gets 2 copies of the recessive allele. Sorry about the overlapping at least text here. I'm going to be honest, it just would have taken longer to fix than to simply say sorry and move on. So ignore those red words that were up there originally, copy the notes you can see, and let's move on. Okay, we are just hitting, looks like 19 minutes on the clock now, and we just have 2 pedigree practices to go. So we're going to get through this pretty quick, and then we're done for today, and done for this chapter, really. So we're going to do this practice pedigree here. It says a darkened shape indicates hemophilia, and a clear shape indicates a person with normal blood clotting. So the affected people in this case have hemophilia, which if you remember is when you can't stop bleeding, you have trouble blood clotting normally. First question says, how many males have hemophilia? So remember, females are round shapes and males are squares. We're going to try to find how many males have hemophilia. Let's just look for the squares with colored in shapes, and I see 1 right here, I see 1 right there, and I see 1 right there. So there are 3 males with hemophilia. Just to mention also, if you see on the left hand side here, little Roman numerals 1, 2, 3, and 4, these are your generations. So this would be a couple up here, and then their kids, and the grandkids, and the grandkids' kids. So I'll leave that there for now. How many females show normal blood clotting? So females, again, are the round shapes, and normal blood clotting would be uncolored. So females with normal blood clotting. I see 1, 2, 3, 4, 5, 6 females showing normal blood clotting. So not too many there. Or sorry, a good amount actually there. How many marriages are there? So how many times do we see a line connecting two people? And really, even that word marriage, really we're saying how many matings are there. It doesn't have to be married, but they did have kids. So right here I see one mating right there. I see another one right here with kids having children, or with people having children. I see one right here with people having children, and that appears to be all. None of these people here are connected with lines through the middle like this, so none of them are mates, they're just siblings. But we do have these 3 sets of parents. So 3 matings, 3 marriages, you want to think of it that way, where people had children with each other. How many children do the couple in row 3 have? So the couple in row 3, there are 4 people in row 3, but the couple, or these 2 on the end, they're actually connected by the horizontal line. And it says, how many children does that couple have? So we're going to follow their line down, and it just now branches off to all these kids in here. All these kids are their children. So it's 1, 2, 3, 4, 5, 6, 7 children for that couple. Question here says now, is hemophilia a dominant or recessive trait? Now, hemophilia is the people that are colored in. Start at the top. If this is a dominant trait, would we expect these children to have hemophilia? Probably, right? And then you'd probably see it here. You'd probably see it down here. I'm going to say hemophilia sure seems like a recessive trait because it's kind of all over the place. It's not necessarily in every generation. You know, it shows up sometimes, but right here, we'd expect that most of these kids would have hemophilia if this parent does. And if this person here had hemophilia, we'd expect definitely at least one of these kids to have hemophilia. So it sure seems like it's going to be a recessive trait. And the last question down at the bottom says, what are the phenotypes of the parents in row 2? So the parents in row 2 are these 2 individuals right here. Remember, phenotype is not what your genes actually are. It's just what you end up showing as a trait. So what is the physical manifestation of those genes? It's asking, do they have hemophilia or do they not have hemophilia? And we know from the question that darkened shape indicates hemophilia. So these 2 individuals right here that are not colored in, they have no hemophilia. They would have normal blood clotting. You haven't figured it out yet? Pedigrees are really just a puzzle. It's really very little difference than something like a Sudoku, where you have to kind of figure out what numbers go where based on what's around and what has to not have conflicts and things like that. You know, if you know some information, there's typically a good amount of other information you can figure out pretty quickly and easily. So we've got another one here, last example. This one says cystic fibrosis is a recessive disorder. This pedigree shows the passing of the cystic fibrosis gene through 3 generations of a family. And we can see from the letters that it's going to be little a's and big A's. So because cystic fibrosis is a recessive disorder, I'm going to write a little note for myself right here that says they have to have then little a. Anyone with cystic fibrosis has to have little a, because a recessive disorder means if you had even one of the big A's, it would overrule the little a's and you'd get a normal affected person. You would not have someone with cystic fibrosis. So anyone with cystic fibrosis is going to have to be little a. So let's try to figure that out. We'll start right there. Anyone I see affected, I now know they need to be little a. So I'm going to try to fill this in because the first question says write in the genotypes on the line next to or below each individual. That means write their letters in. So I'm going to start right at the top with person 1, and I know since they are colored in, they must be little a, because they are affected. That's the only way they're getting this recessive trait is if they have only 2 copies, or sorry, a whole 2 copies of the little recessive trait. Same thing with the female at the bottom here. She's also going to be little a. You can see up top, person number 2 has 1 big A, 1 little a. Even 1 copy of the big dominant gene rather, means they do not show the trait. They would have to have 2 little copies like person number 1. Back down to the bottom, people 11 and 12 are also going to be little a, because to show the recessive trait, they only have that option. Let's try to figure out the rest of them now. For these kids, Let's start with kid 3. What genes can the parents give? I see right here the dad. The only option he's got are little a. So he had to have given a little a to person 3, person 4, person 5, person 6, person 7. Do any of them have the recessive trait, though? Do any of them show cystic fibrosis? None of them are colored in. None of them are affected, which means they had to have at least one uppercase A. So a big A here, a big A here, a big A here, a big A here, a big A there. Every one of them has to have at least one copy of the big A because they do not show the trait. And really, that should have been the first thing I did probably. Because now I can go back and say, well, does it matter what the second allele is? Not really. You know, they could have a little a, they could have a big A. We don't necessarily know, except that we know the dad can only give a little a because that's all the dad has to give. So every one of them now, person 3, person 4, person 5, person 6, person 7, has to be big A, little a, because they had to have gotten a big A from someone to not show the trait. That had to have come from the mom up here because she's the only one that has the big A, and the dad can only give little a's. And that's it. That's everybody. We filled in everybody's stuff. This person number 7, the son, big A, little a, mates with a woman with 2 big A's. So the kid gets 1 big A from the mom and 1 little a from the dad. And then person 10 at the bottom was also filled in. So this last question here, why did individuals 9, 11, and 12 get the disease, but their parents did not have the disorder? So why did 9, 11, and 12 get the disease even though their parents here and their parents here did not have the disorder? And that's because their parents are carriers. This mom and this dad each had a little a. They didn't show the disease themselves because they each had 1 copy of the dominant trait. And this dad and this mom, the same thing. They each had a heterozygous big A, little a genotype. So even though they don't show the trait, this little a, this little recessive gene is hiding in their genome. It's hiding in their DNA. And there was a 50% chance it would show up right here. Each of them just dumb luck happened to give the little a to their daughter. And these people over here, it happened twice, but they happened to have given the little a's to each of their daughters there. That is the end of Heredity. Don't look now. We've got 1 chapter left. Next week, I expect, I said before, we'll probably do some kind of lab or activity or something for one of the days, not jump right into the next chapter. I wanted to try to do something fun, or more fun than this stuff at least. Go now, finish the forum for today. And you got some worksheets posted to OneNote. That'll be due on Tuesday. Now they're doing the new schedule. Stuff due Tuesday instead of Monday. I'll have office hours Monday at 10 a.m. and then extra help Tuesday at noon before, once again, work is due that night. So that's all for now. Have a good weekend. Look out for each other. I'll see you in a couple days.